Hi everyone, my name is Kezley and I did my research on measuring ADH activity and ALDH gene expression in ethanol exposed CACO2 cells. Alcohol use disorder affects many people in modern Western society. Understanding how our body reacts to alcohol can give rise to more information on this topic and can give rise to information on what can be done to help others. Two enzymes are involved in alcohol metabolism, with the first one being alcohol dehydrogenase, ADH, and aldehyde dehydrogenase, which is ALDH. ADH converts alcohol to toxic acid aldehyde, while ALDH converts the toxic acid aldehyde to acetic acid. By applying alcohol to CACO2 cells and analyzing the enzymatic activity and gene expression can give us further information on this topic. We hypothesized that the ADH activity and gene expression would increase as the ethanol treatment amount increased. An MTT assay was first performed to determine the sublethal dose of ethanol, followed by the ADH activity assay on a high and low dose of ethanol, which was determined by the MTT assay, and then the gene expression of ALDH1B1 on both the high and low doses of ethanol on the CACO2 cells was measured. The first result that was found was the increase in cell metabolism at 2% ethanol-treated CACO2 cells, while they were significantly lower metabolism at 5%. Both of these doses were used for further tests. The second result that I found was the alcohol dehydrogenase enzymatic assay, which displayed no ADH in the untreated 2% treated or 5% treated test samples. The positive control was significantly higher compared to the other samples, meaning that the assay had worked, but the samples did not release ADH, or the amount of ADH that was released was not detectable in this assay. The third and final result, the PCR results on ALDH1B1. We found that there was no significant change between each group compared to the untreated CACO2 cells, but there was a trend in the positive direction for the 2% treated CACO2 cells, which could be due to the MTT assay having high cell metabolism at 2%. In conclusion, we showed that treating CACO2 gut cells with ethanol increased metabolism at 2% and decreased metabolism at 5%. CACO2 cells with these ethanol concentrations did not release ADH and did not change the ALDH1B1 gene expression significantly. This study can be further explained with more research being performed. The range of ethanol that was used in the MTT assay can be further researched to narrow it down to a more specific amount of ethanol that kills the cells. The gene expression of ALDH2, another common gene that is involved in ethanol metabolism, can be tested instead of the ALDH1B1 to compare results. Also, the whole study can be repeated using HepG2 liver cells, as the liver is the main organ for cell metabolism. Overall, these results can give more detail on how the body reacts to alcohol consumption and therefore alcohol use disorder.